So good afternoon, students. So we'll be having a lecture on. Yesterday we had a lecture on introduction to forensic science, right? First we'll brief about that. We'll just brief about yesterday's lecture. That is what we learned about the definition of forensic science as an applied science that uses the philosophies of all other natural sciences to aid the criminal justice system. Then we saw the forensic triad. Then we got with the principles of forensic science. There were 7 principles of forensic science or laws of forensic science. The first one was principle of individuality or law or individuality, where everything which is man-made or natural has their own unique properties. Second one was mutual exchange. Every contact leaves a trace. Okay, second one was mutual exchange. Every contact leaves a trace. That is given by Edmund Locard, and he was known as the father of modern forensic science, Edmund Locard. And the third law was law of progressive change, that stated that everything changes with the passage of time. The criminal, the crime scene, the evidence, everything changes with the passage of time. Fourth one was comparison. So only likes can be compared. That means if you are comparing something, one sample, the origin of the other sample which we are comparing it to should be the same or similar. The other one was, the fifth one was principle of analysis. That just means that when you are collecting a sample or when you are transporting it, when you are analyzing it, you have to follow the standard procedures. Okay, you must not deteriorate the sample. The sample collection has to be perfect. You should use the standard procedures for collection. Sixth one was law of probability. Sixth one was law of probability, where law of probability was just a mathematical concept where it shows the assumptions that we make in a crime scene. The last one was law of circumstantial fact. That just showed that men lie but evidence don't. So it showed that we should not depend totally on the evidences what witnesses get, testimonies. We should try to focus on the evidences like factual evidences, like the evidences which we get from crime scene. Now we will go on to today's lecture. Today's lecture will be about the history of forensic science. Okay, history of forensic science. Now we'll go the forensic in the world, then we'll come to India. Okay, we will finish the world first, then we'll come to India. So yes, yesterday we had discussed about the first case, right? The first one was in the 12th century in China. There were written book known as the washing away of the wrong, and the author observed that in lungs of drowned victim water got collected. Okay. It also mentioned about strangulation. I told you what is strangulation yesterday. It also mentioned how strangulation can affect the cartridges of the neck. Okay. This was water collected in lungs. You saw it yesterday. I explained you the side yesterday. Now we will go forward. Yes. So the earliest record of applying forensic science, in the criminal investigation was in China. Okay, that also was in China. That was around the 3rd century. So you have a manuscript, a collection of criminal cases. Okay, where how a coroner solved a case claiming of death due to accidental fire. So coroner means just the one who is doing the postmortem. Okay, in India we call the surgeon or something postmortem, the one who does postmortem, but in other countries they call them as coroners. Okay, so the case are that like a man was burned. Okay, man was burned to death. And the wife was a suspect. So what the coroner did was he took the body and he checked. Okay, he checked and he found out there was no ash in the mouth of the man. There was no ashes present inside the mouth of man. So he inferred that if he was burned alive, he should have inhaled some ashes also, right, while burning. That should have been in his mouth, but it was not present in his mouth. That means that the man was already dead and they burned the body afterwards. Okay, so that is how they implied first forensic science as a criminal. For a criminal capture, they firstly applied forensic science like this. So that was in China in the 3rd century. So the coroner performed experiments. We cannot just tell, like make assumptions, right? As a forensic scientist, we have to conduct some experiments. Okay. For example, there was ash in that man's mouth, so we must know that whether ash will be present in the body that was burning. So we must know that. For that, we must conduct experiments. Like in humans, we mainly, for comparison with humans, we mainly do experiments on pigs, okay, because pigs has less hair also, and their fat content is almost similar to human beings. That is why whenever we want to like experiment on something related to human, we use pigs. Okay. So the coroner, what he did, he burned 2 pigs, okay, and he checked. One pig was already dead, the other pig was alive. Okay. So the already dead pig, he burned it, and he burned the other pig alive. Okay. And he found out that the pig which was burned alive had ashes in his mouth. Okay. And the pig which was already dead and burned afterwards did not have ashes in the mouth. That is how he solved the case. He told that this was the reason that man doesn't have ash in his mouth, so he may have been dead and then burned. Then the wife had to confess. She confessed the crime, that she killed him, then he burned him. Okay. Okay. The next one was about fingerprints. Okay. The next one was about fingerprints. So in 1686, whenever you are history can be boring, but you must learn it. So just note the dates. Okay, note the dates and the persons. You will be having a notebook, right? So just note the dates and the name of the person when I am telling, and what is his contribution. So in 1686, there was a person called Marcello Malpighi. Marcello Malpighi is a professor. Okay, he was a professor. He recorded notes about fingerprints. He was the first person to record something about fingerprints. Okay, he recorded some notes about fingerprints. He told that he had a treatise. Malpighi noted in his treatise ridges, spirals, and loops in fingerprints. Treatise means written records. Okay, in his written records, he told that fingerprints had ridges, spirals, and loops in them. So he first told you, as a forensic scientist, you must observe. Okay, Malpighi observed the fingers, and he found that these patterns on the fingers, they were in some kind of spiral patterns were there. There was some kind of raised pattern. Those are known as ridges. Some things were raised. Okay, something went in loops. So he observed that, but he didn't give any classification or something. He didn't make it as an important thing or something. Okay, that was his main loss, or else he would have been the father of fingerprints. He just told that this was there, that's all. He didn't stress it upon its importance. He didn't look after that. However, he did not acknowledge the value of fingerprints as a method of identification. If he would have acknowledged its value, that these are unique to each person, if he would have done some more research on that, he would have been the father of fingerprints, but he didn't. He just told it was there, that's all. So, but a layer of the skin was named after him. Okay, a layer of our skin was named after him. That layer is known as Malpighi layer. Malpighi layer. So this was Marcello Malpighi. So I have added the images. Okay, I have added the images. So just visualize this. Just because in some exams you may have some image. Mainly in quizzes. During quiz they will show you an image and identify this person. Okay. Must have been a contributor to forensic science. So just identify this person. They will just ask you like that. So you must, whenever you are, I only mentioned the important persons in forensic science, and I have put their images also. So you can easily identify them. Okay, this was Marcello Malpighi. Yeah, Malpighi, because of his contribution, a layer of skin was named after Malpighi. It is known as Malpighi layer. So check your hands. When you check your hands, you can see the raised patterns on the palms, right? You can check your hands. There's a raised pattern on your palms, right? That pattern, there's ridges, there is some kind of dip like a hill and valley. Okay, that layer, that full layer is known as the epidermis layer. Epidermis layer, and underneath that there's a dermis layer. So this epidermis layer, the first layer, is also subcategorized. It is subcategorized into stratum corneum, stratum lucidium, stratum granulosum, stratum spinosum, and stratum basilae. You don't need to study deep about this. I was just telling you the layers. And Malpighi layers mean the stratum spinosum and the stratum basilae together are known as Malpighi layer. We will learn more about the layers when you are learning fingerprints. So just for your common information, just to show you what is an LPG layer, I included this. Okay. So, stratum spinosum and stratum basal layer together are known as LPG layers. Yeah. So, the next one was in 1775. 1775, there was a Swedish chemist known as Carl Sechley. Carl Sechley, he devised the first successful test for detecting the poison arsenic in corpses. So, in the olden times, the main way to kill some person was poisoning. It's very simple. Nobody knew how to find the evidences. Okay, you would mix something in the glass. Cyanide was mainly used. Arsenic is a metallic poison that was abundantly used in the 17s and 18s, the old eras. So nobody would know, right? Nobody would know what happened. They didn't have the internal autopsy, postmortem. They didn't know about all this. So it was easily used. We could not identify who did the crime. Okay, so the first one was in 1775, Carl Sechley devised a test for detecting arsenic. He devised the first test for detecting arsenic. Okay, you won't need to learn about the test. You will learn about the test and in toxicology. Now just history. That's all. Okay. So he devised a test. Then the most significant early figure in this era. So in the field of this testing, chemical testing or toxicology, you call it toxicology. So the most significant early figure in this area was known as Mathew Orfila. Mathew Orfila, he was a Spaniard, he was from Spain, okay, who is considered as a father of toxicology. Now we are coming to the main people. So Mathew Orfila is considered as the father of forensic toxicology. Father of forensic toxicology is known as Mathew Orfila. In 1814, Orfila published some scientific treatise on the detection of poisons and their effects on animals. See, in 1814, Mathew Orfila published a book. Treatise means just publications. Okay, he published that showing there are so many poisons and their effects on the body. Okay, their effects on animals. Mainly at that time they were not using with humans, right? They used to study on animals. This treatise or this book established the forensic toxicology as a legitimate scientific endeavor. So Mathew Orfila's book, okay, established forensic toxicology as a legitimate scientific endeavor. Okay, scientific toxicology became a subject from there. So he was considered as the father of forensic toxicology, Mathew Orfila. The date is 1814. This was Mathew Orfila. You must identify Mathew Orfila's feature. I told you in quiz exam, when you are conducting quiz and they just present you the picture and identify this personality, Mathew Orfila. Okay. Yes. Now we left the 70s. Now we are going to the 1800s. Okay. So in 1828, William Nicol invented the polarizing microscope. So there was something called polarizing microscope. This person invented it. Not that significant. Okay. Like we learn about microscopes later. You learn about polarizing light. You would have known all this, right? 12th physics. We learn about that later. So he invented the polarizing microscope. Then the other one was in 1853. 1853. Dr. Tieckmann gave the first microcrystalline test for hemoglobin. Microcrystalline test. The test is known as Tieckmann's test. Tieckmann's test. So microcrystalline test. So what happens? There is hemoglobin in blood, right? So when you are doing a blood test, to find out whether it is blood or not, we use this crystal test. This is still used. Tieckmann test is still used. The two crystal tests that we still use is known as Tieckmann's test and Takayama test. You will learn that in biology. But in 1850, this was the first microcrystalline test. Okay, that is why it's important. 1853, Dr. Tieckmann. Okay, he invented the first microcrystalline test to identify whether the substance was blood or not. Microcrystalline test means, for example, if you use that reagent, Tieckmann reagent, it will combine with hemoglobin, it will form a crystal structure. Okay, this crystal structure you can put under the microscope. It has a definite color and a definite shape. Okay, so it can identify blood. You will have the practicals, then you will understand it more. Just understand. Tieckmann invented the test, microcrystalline test for blood in 1853. Okay. Then after that, German scientist Christian Schauben gave the first presumptive test for blood. So Tieckmann's test was known as confirmatory test, and Christian Solzman's test was a presumptive test. So when you are like testing for blood, you use two tests, like salt analysis. You had salt analysis in your 12th standard, right? Chemistry. So what do? You have a presumptive test and a confirmatory test. Okay, just like that, blood also have presumptive test and confirmatory test. There are so many presumptive tests for blood. There is like phenolphthalein. There are so many tests. Okay, phenolphthalein, peroxidase test, so many tests. And Tigman's test was a confirmatory. Crystal test was a confirmatory test, and what Chauvin gave was a presumptive test. So, yeah, in 1839, toxicological evidence was first used in trial by a Scottish chemist named as James Marsh. He testified the detection of arsenic. So, for example, if you are a forensic scientist, your job is not to just analyze the sample. You will have to go to the court of law. You have to stand in the court and you have to explain that. Okay, you have to explain the scientific principle behind the death. Okay, judge may not know the test. He may not know. He may be not from a scientific background, right? So you are the person who is responsible. You must go there and you must tell, sir, this is how he died. These were the symptoms that I found on his body. This may be due to this poisoning. Okay, like that you have to explain. As a fingerprint expert, you will go, so these were the patterns that I found. I compared these many parts of the fingerprints, and according to the standard rule, if you compare this much parts and that is matching, then it is almost similar fingerprints. That is way you have to explain to the judge. Okay, then only you will win the case. So first toxicological evidence testimony was given by James Marsh. He told that the death was due to arsenic. He gave the testimony in court. The first court testimony in toxicology evidence was given by James Marsh in 1839. Okay. Yes. Now another important person in forensic science, Alphonse Bertillon. So Alphonse Bertillon. So in 1879, Alphonse Bertillon, 1879, Alphonse Bertillon applied anthropology and morphology for personal identification. Mainly study of bones is anthropology, okay, and morphology. So I will tell you what it is. So he merged these anthropology and morphology into one system. He called it Bertillon's system of anthropometry. He called that system Bertillon's or Bertillon's system of anthropometry. Okay, it was known as anthropometry. He merged anthropology with morphology. Okay, so Bertillon's system of anthropology was a procedure that involved taking a series of bodily measurements and means of distinguishing one individual from another. For example, what it means is he used body measurements. So there was nothing to individualize a person. So what Bertillon used was, he used anthropology and morphology. He mixed them together and he formed something called his own science. He formed that is known as anthropometry. So in anthropometry, for example, take an example of me. Okay, they will measure my height. Okay, they will measure by the distance from the sockets. Okay, distance from the ears. Okay, fingers, how much distance my elbows are. Okay, joints are. All those physical features they used to measure and they used to record. So he considered that as very individualistic. Okay, for every person that will be different, right? So it was individual, considered individual, but it was time-taking process. You have to measure everything, right? It was a time-taking process. For nearly 2 decades, for like 20 years, the system was considered the most accurate and precise method. Okay, for almost 20 years. So what happened after 20 years? We'll get to that. So for almost 20 years, this Bertillon's anthropometry was the most accurate system for identification. So his efforts, Bertillon's effort, earned him the title of father of criminal identification. Bertillon's anthropometry earned him the title of father of criminal identification. Bertillon was known as the father of criminal identification. So 1879, Alphonse Bertillon, anthropometry, known as the father of criminal identification. Fine. You will encounter Bertillon in so many slides. Like this was Alphonse Bertillon. It was a person, Alphonse Bertillon. Okay. This was anthropometry. Okay. Anthropometry. You will take the height. You can see the distance from one hand to other hand. Okay. When you are sitting position, how much is your height? Then they will take you ear to ear. Okay. Standing position, they'll take the feet, chin, this part, everything, fingers, everything. Bertillon used to make a record, and that is how he used to individualize. You know how much time taking processes will be. Okay, so but almost 20 years, this is the most accurate method for identification. Yes. Now after 20 years, what happened? Fingerprints came in. Fingerprints, till now, fingerprints is the most accurate method. Okay. Before that, anthropometry was, but just after 20 years, fingerprints came, and that was the doom of anthropometry. Nobody else, because time taking. Anthropometry was very time taking. Fingerprints was very simple. You could just give the prints, you could identify. Okay. Till now, fingerprints is it, the identification. You have mobile phones, everything, fingerprints. So in 1880, Dr. Henry Falls published an article in the scientific journal Nature. Nature is a big scientific journal. You can search for it afterwards. Scientific journals. You just finish your 12th. I will be coming into BSc and MSc. Then you will have to publish your research works. You will be publishing in all those journals. Okay. You will get to know in the later courses. Okay. No more book feed, nothing. You will have to do your own research. You will have to study. You have to make your own research papers. So interesting. BSc and MSc will be very interesting. So Henry Falls published his research paper in Nature, and he discussed fingerprints can be used as a identifying feature. Okay. He was also credited with the first latent print identification. Okay. He was credited with the first identification using fingerprints. He also told that you can identify people using fingerprints. Okay. That is what Henry Falls did. He published his published his paper in the journal called Nature. That was in 1880. In 1892, Francis Galton, there was a person called Francis Galton. Okay, he published a book. Okay, what is bigger than a journal? A book. Okay, so Francis Galton published a book titled Fingerprints. Okay, titled Fingerprints, which contained the first statistical proof. He statistically proved that fingerprints were can be used as an evidence. You can identify individuals using fingerprints. All those things Galton wrote in his books. His book was known as Fingerprints. Okay, and he published it in 1892. Okay, his book went on to describe the basic principles that would form our present system of identification by fingerprints. That book is still the basics for fingerprint identification. Okay, Fingerprints by Francis Galton. So this was Henry Falls, Sir Francis Galton. After his phenomenal work in fingerprints, he was given knightship by the British. So he became Sir Francis Galton. Okay, there were conspiracy theories in fingerprints. There were theories the theories telling that Francis Galton took the work of Henry Falls. Okay, there are so many theories. But still, Francis Galton is considered as the father of fingerprints. Okay, father of fingerprints, Sir Francis Galton. Okay, there are other theories you will get to know. We will teach you all the theories while you are dealing with fingerprints. Okay, for now, just focusing on the history. So this was Henry Falls. That was Sir Francis Galton. Okay, 19th century. So 17 is finished, 18 is finished, 19th century. Yes. So ironically, in 19th century, there was not a person. It was not a person who was the pioneer of forensic science. It was a character. It was a comic character. He was known as Sherlock Holmes. Everybody would have known Sherlock Holmes, right? You would have watched his series. You would have read the books, right? So ironically, the best known figure in the 19th century forensics is not a real person but a fictional character, the legendary detective Sherlock Holmes. When it comes to forensics, everybody thinks of Sherlock Holmes. Okay, if you have not watched Sherlock Holmes, watch Sherlock Holmes. It's better to read Sherlock Holmes. Okay, there are 3 books. You can read Sherlock Holmes. You'll get it on Amazon. Buy it, read it. Okay, it's very good. Start cultivation of reading. Okay, start reading. Read Sherlock Holmes as your basics. Read Sherlock Holmes. So Sherlock Holmes was a fictional character. Who created Sherlock Holmes? Sherlock Holmes was written by Sir Arthur Conan Doyle. Arthur Conan Doyle wrote Sherlock Holmes. His novel was Sherlock Holmes. Okay. So that novel popularized the methods of scientific methods in criminal investigation. using science in criminal investigation. That his books popularized it. Okay. Holmes was the first person to apply the newly developing principles of serology, fingerprints, firearm identification, and question document examination long before their value was recognized and accepted by real life criminal investigation. See, Sherlock Holmes used forensic serology, fingerprinting, firearms identification, question documents, all those things. Think we have not even, we only covered fingerprints right now. Serology is remaining, question documents is remaining, ballistics, firearms is remaining. So, in the Sherlock Holmes, that book, he covered all these things. History didn't cover till now. We are just going to serology and ballistics afterwards. Okay. So, that is considered like beacon, as the beacon of criminal investigation, Sherlock Holmes. So, that is all about the 19th century. Yes, Sir Arthur Conan Doyle. Okay, he was Sir Arthur Conan Doyle, the writer of Sherlock Holmes. Yes, now we are coming to the present century, the 20th century, the decade before. Now we are moving on to the 21st century, right? So, that's the last century, the 20th century. The main things were blood grouping was invented. Dr. Karl Landsteiner, you would have read about him in biology, right? Your biology book, 12th biology book, you would have read about Karl Landsteiner. So, in 1901, Dr. Karl Landsteiner discovered that blood can be grouped into different categories. Okay, it was A, B, AB, and O. Okay, so Landsteiner, till that time, they will only use that this was blood or not. Okay, they didn't know how to group the blood. If you get the blood from a crime scene, now you can group it, you can identify DNA. Methods are very high. So, before the 20th century, they could only identify it as blood or not. Okay, so Landsteiner gave discovered that blood can be grouped into A, B, AB, and O. That was in 1901. So, Landsteiner is thus considered as the father of blood grouping. So, we have another, that is Karl Landsteiner. He was considered the father of blood grouping, 1901. Yeah, in 1915, Dr. Leon Lattice devised a relatively simple procedure for determining the blood group of dried blood in a blood stain, a technique that he immediately applied to criminal investigation. So, in 1915, there was a doctor called Leon Lattice. Okay, so in crime scenes, when you go to crime scenes, blood won't be always wet, right? When you have a cut, blood will dry off. Okay, so it was hard in those times. They didn't know how to collect this and identify this. So, blood grouping is usually done. You prick the finger, you get the fresh blood, then you put the reagents and you do blood grouping. But what if it's a dry blood? Okay, usually in all the crime scenes you will get dry blood. So, how? Leon Lattice, Dr. Leon Lattice, 1915, he devised a method to collect the sample and identify the dried blood. Okay, so that was developed by Leon Lattice. He used that for criminal investigations also. You will learn all the methods, don't worry, you will learn all the methods in biology. Okay, now just focusing on history. This was Dr. Karl Landsteiner. Yes, now 1910. First one was 1901, now it's 1910. Albert S. Osborne wrote the first significant text in the field of question documents. This book is still a primary reference for document examiners. There is a field called question documents, like for example handwriting, signature, whether it is. So question documents means any documents or handwriting or something which their legitimacy is challenged. Okay, we don't know if it was a legitimate person who did that. So we send those things to the question document department in a forensic science lab. They are the people who analyze it. So the pioneer of question documents was Albert S. Osborne. Albert S. Osborne, his book question document is still used. His book named question documents is still used. Okay. Osborne's development of fundamental principles of document examination was responsible for the acceptance of documents as a scientific evidence by court. Osborne is therefore referred as father of question documents. So, because of Osborne, new science in forensic was formed, that was question documents. Osborne gave his book on question documents. So they were later considered as a significant evidence in court. That is why he is known as father of question documents. Albert S. Osborne. Date was 1910. Okay. Yes, this is Albert S. Osborne, father of question documents. Question, not question, Q-U-E-S-T-I-N-E-D, question documents. Yes, another important contribution in forensic science, that is in the 20th century, early 20th century, by Frenchman Edmond Locard. You must know who is Locard by now, right? Exchange principle. Okay, he was the pioneer in forensic. Okay, he was known as the father of modern forensic science. So, Frenchman Edmond Locard. Locard asserted, yeah, this is the principle of exchange written here. That is, when two objects come in mutual contact, they will exchange something. Okay, every contact leaves a trace. Locard gave the basic principle of forensic science. He is therefore referred to as the father of modern forensic science in 1910. So, Locard was like he was all into forensic science. So, he was in France. So, he wanted his own department of forensic science to use science in crime. Okay, he was very motivated in that. So, in 1910, he persuaded the France police, Lyon's police, okay, to give him 2 rooms in their attic. So, the police gave him 2 rooms. From there he started the first criminal laboratory. Think the first criminal laboratory was started by Edmond Locard in Lens, France. Okay, Lens, France. That was in 1910. He started the first forensic laboratory, first criminal. Yeah. In 1910, he persuaded the Lyons Police Department to give him 2 attic rooms and 2 assistants to start a police laboratory. Okay. It served as one of the first criminal laboratory in the world. It served as one of the first criminal laboratories in the world. That's all about Edmond Locard. You should learn the picture of Locard. Okay, father of forensic, father of what you are studying now. So, Edmond Locard. Yes. So, coming to 1920, there was a person called Colonel Calvin Goddard. Okay, this colonel called Calvin Goddard. 1920. So, he refined the techniques of firearm examination by using comparison microscopes. So, what Goddard did was, there were so many cases which he investigated. So, whenever you fire a firearm, okay, the bullet goes out, right? There is a pattern that bullet will have from the firearm. Okay, this is known as rifling pattern. You will learn more about in ballistics, but every gun, when you are firing it, there will be a pattern. Okay, that bullet, you will get the pattern of that gun. So, if you are having 2 same bullets fired from the same gun, then you can compare them easily. Comparison microscope. Okay, you can keep them in comparison microscope, easily you can compare them. Okay, that is all about ballistics. Okay, so Colonel Calvin Goddard did that. First comparison, like fingerprints, you can compare bullets also. Okay, he was a pioneer in examination and identification of firearms and bullets, and is known as the father of forensic ballistics. Okay, so there's another person. 1920, Colonel Calvin Goddard, father of forensic ballistics. Okay, Goddard's work allowed investigators to determine whether a particular gun has fired a bullet by comparing the bullet with another that is test fired from the suspect's weapon. Okay, his expertise established the comparison microscope as an indispensable tool for the modern forensic firearm examiners. So, till now we use the comparison microscope for firearm examination. When you go to a ballistic department, the first thing you will be seeing is a comparison microscope. Okay, till now. He was the person who used it first. Okay, he made that comparison microscope come into forensics by examining bullets. We can use comparison microscope for many other examinations. You can examine hair, okay, all those things. But when you are telling about comparison microscope, it's mainly concerned with bullets and ballistics. Yes, this is Calvin Goddard. This is the comparison microscope. You may not be familiar with comparison microscope. You must have used compound microscopes in your biology, right? Botany, you will be having compound microscopes. So, this is comparison microscope. You have 2 stages. You can keep 2 samples and simultaneously you can visualize the samples A and B just like this. You can put them on top of each other, or superimposition is also possible in comparison microscope. Okay, that is how we can identify it. Yes. So the most significant advance in forensic science in the 20th century, though in our 20th century, 20th century was by Alex Jeffries. You would have heard, right? Alex Jeffries, Dr. Alex Jeffries or Sir Alex Jeffries, in the field of DNA profiling. DNA came into forensic in 1984. 1984, Sir Alex Jeffries DNA profiling. He did the first DNA profiling. You can read about the case. Please read about the case. Okay, I want to explain the case now. Please read about Colin Pitchfork case. Colin Pitchfork case. Please read about it. It's very interesting case. In forensic, you need to learn about cases. Okay, start cultivate the reading of like case studies. Start reading case studies. Okay, so in 1984, Sir Alex Jeffries brought in DNA fingerprinting. Okay, that was like the till now we use DNA fingerprinting only. First thing about when you hear about forensics, it'll be like first is fingerprints and second is DNA. Okay. Combined with the modern analytical tools, DNA typing has revolutionized the practice of forensic science. So we will end here, like DNA was the last. He is still alive, he is living in our era. So we put a color photo of him, that is Sir Alex Jeffries. Okay. So we'll just refresh the slides. What we have learned today. So we have learned about the history of forensic science in the entire world. Okay. First one was 12th century, that is in China. There are so many other historians about before this. Okay, I told you Archimedes also was considered as the father of forensic science. Some historians tell like that. Okay, but the proof, definitely we need definite proof, right? So first was in 12th century, that is Chinese. He attempted the book was The Washing Away of Wrong. Okay, where they told that water was collected in the lungs of the dead person. They also told strangulations can be identified by checking the cartilage. Then we go to the 3rd century. Collection of Criminal Cases was the book. Okay, that case where I told ash was present in the mouth, that case. Okay. Yes, then we'll go to Marcello Malpighi, 1686. Okay, he told that fingerprints, they had some kind of loops and patterns in them. That was given by Marcello Malpighi. Okay, he didn't stress too much about the importance. He was given one layer of hand was named after him, the epidermis. That layer is, in the epidermis layer there are two layers, stratum spinosum and stratum basale, together known as Malpighi layers because of Malpighi's contribution. Then we have, then the important person is almost in 1814, Matthew Orfila. 1814, Matthew Orfila, father of forensic toxicology. Okay. 1800s, then we have Tigman, Dr. Tigman, 1853, first microcrystalline test for blood. 1879, Alphonse Bertillon, father of criminal identification. The science was the anthropology together with morphology known as anthropometry. Okay. 1880, Henry Fowles published in Nature was the journal's name, published in Nature about the importance of fingerprints in crimes like criminal identification. 1892, Francis Galton, our father of fingerprints. Okay. 19th century, Sherlock Holmes or Sir Arthur Conan Doyle. Then we'll just refresh through 20th century. 1901, we have Karl Landsteiner, ABO blood grouping. Leon Lattice, 1915, he used the dried blood for grouping. 1910, Albert Osborne, father of question documents. Edmund Locard, 1910, he started the first criminal identification laboratory, Lyons, France. 1920, Calvin Goddard, father of forensic ballistics. And in our present generation, we have great Sir Alec Jeffries, 1984, DNA fingerprinting, father of DNA fingerprinting. Okay. So we'll end here. Refresh the history. History is a little boring, but you have to study the history. So refresh the topics. Tomorrow we'll try to learn about the Indian history. Okay, we'll learn about departments. First we'll learn about the departments, then we'll learn about the history. That will be easier for you, because if I am telling about the history, there will be different departments involved, all right? So we'll learn about the departments, then we'll discuss the Indian history. Okay, thank you.